Hello, everybody. We are now in week 8 of our course. So this is the second part of microbial nutrition and growth. So here we go. Just so you know, chapter 5 is the viral growth and chapter 6 is microbial growth. So you may have to go back and look at some parts of chapter 5, and there may be some parts that I covered today that will be review of part 5 or chapter 5. So here we go. Okay, so when we think about the growth of bacteria, remember how we said that essentially one cell grows to make two. So for those of you that's had a class and you talked about eukaryotic cell division, it's not that much different in thinking about one cell literally forfeits itself to make two identical cells. So the problem being, or the challenges being with bacteria, is they don't have all those subcellular membrane-bound organelles, nor do they have chromosomes as we know them in eukaryotic organisms, and they don't have a nucleus. So in actuality, the division of bacteria should be pretty straightforward. So we call it binary fission, so simply splitting into two. And it's not very surprising how this happens. You have one cell, it's going to grow larger, it's got to get more cytoplasm, it has to have more ribosomes, it has to have those materials to make the two new cells. Now, when the two new cells are made, they're not going to be as big as that original cell. So sometimes we will call that original cell the mother cell, and then we'll refer to the two cells that's made a result of that one cell, the daughter cells. So when we think about the bacteria chromosome, so we're essentially thinking about this circular piece of genetic material. And essentially what it's going to do is it's going to replicate itself, and it will attach to the inside of the cell envelope. And as that cell begins to pull apart, there is going to be I believe it is a carbohydrate-based septum or wall that's put between the two new sources of material in the daughter cells. And then we have essentially the cutting off of the two cells. So let's take a look at the figure. You might remember I tried to help you all understand like outside in and inside out. So that's where this helps in your thinking when you're trying to visualize binary fission. So remember, inside of our cell, we have that blue background layer. That's the cytoplasm. There is a membrane around it. So the cytoplasmic membrane or the cell membrane or the plasma membrane are all acceptable terms. And then just outside of that plasma membrane, then we have the cell wall. Now, as you all know, that can have many parts and many different chemical components, but that's in a previous chapter, and now we want to focus on binary fission. So this shows us a bacillus bacterium, and you see that essentially that wad of circular DNA is going to replicate itself. And essentially then it doesn't really show it being attached inside the plasma membrane, but it is. And then as the cell elongates, then we have this septum that is going to put down between the two areas. And I was mistaken, it is protein, not a carbohydrate. It's a carbohydrate in plants, actually. They do something very similar. So we have that wall put down, and then eventually the wall pinches in, and so you just have what we call a septum. It kind of looks like a belly button in between those two cells. And then when that septum is complete, so essentially you can imagine that it's becoming part of the cell wall material this whole time. So we have the division of the two cells. So now we have two new cells. Now notice the cells here look to be the same size as the original cell, but that's not always the case, simply because it takes a little bit of time for that cell to get all the other things that it might need. All right, so we want to think about bacterial growth. So when I think of bacterial growth, I often think about the situation in our house where, you heard that scratching inside the wall and you said, nah, I don't really want to deal with that today. And maybe it's nothing. And then the cats catch a mouse, right? And then you're like, if a mouse is actually coming out to where there are cats, there's probably not just one mouse. So you probably heard that mice reproduce exponentially. So you really don't want to be ignoring, any number of mice, any evidence of mice that you might have in your home. So let's go back to bacteria. When we think about bacterial growth, we're thinking about a couple of things. One of those things is generation time. So generation time is essentially the time it takes for that population to double. Okay, so we have some examples here that Escherichia coli in your colon or in your large intestine is going to be doubling every 20 to 60 minutes. So if you end up with 1, then after that time period, there's 2. That time period passes again, there's 4, 8, 16, 32, 64, and so on. You can see the Mycobacterium tuberculosis, which is the bacterium responsible for TB, has its doubling time as 15 to 20 hours. Go on, you can see the bacterium responsible for leprosy has a much longer generation time. So most of our pathogens have a relatively short generation time. This is how they can cause problems very quickly. Certainly those bacteria that have long generation times may just be, I'm going to say, festering under the radar. So it takes longer for the symptoms to occur, but when the symptoms do occur, they can be worse. So the generation is when it increases the population by a factor of 2. So there's a little formula that we can look at in just a minute. And it says as long as the environment remains favorable, so there's enough food, there's enough space, temperature is great, all the wonderful, Goldilocks conditions, as I call them, then certainly the bacteria would continue to have this doubling effect over time. All right, so this slide, mathematics of population growth. So essentially on the left-hand side, we're just seeing where someone has diagrammed out, essentially you see these vertical lines or columns. And in those columns, it's showing you how many number of cells are present at that time. So in the first true column, there's 1 cell. So that's our mother cell. And then you can see in the second column, there's 2. Third column, there's 4. So we see this doubling until we get to 32, and it continues and continues, right? So the generation time. The mother cell is really generation 0. She is, it is the founding cell. So generation 1 would be the results of that mother cell dividing by binary fission. So the number of generations 1 is going to equal 2 cells. So then we can see, okay, if we know that there's this doubling rate, then we can take the doubling number, which is 2, and we can increase that by the exponent of which generation time it is. And so when we do that, we can easily calculate it. I don't have to do that doubling in my head like I was earlier. Now, if we take these numbers and we plot it here in graph C, you can see that here in figure C, you can see that if you plot the actual numbers of cells on this, the x-axis is time, the y-axis is the number of colony-forming units, which essentially is the number of cells, then you can see that it illustrates an exponential rate of growth. So numbers can remain low until those numbers get high, and now we're doubling bigger numbers. So hence, the more cells you get, then the more growth you're going to have. So that's what's going to happen over time. Now, if we take a look at figure B, this essentially is taking log paper. So we are now just looking at essentially the growth of that bacterium, looking at it more directly, I guess. And you can see that it makes this straight line. So essentially we're seeing that exponential growth, but we are standardizing, if you will, for log. So when we do that, we take away thinking about it geometrically and doing it logistically, then we can see that there's this exponential rate of growth, and the line is literally a sharp line. Okay, so you should be able to calculate the number of cells present for a culture with a specific doubling time. So I promise you won't have very many mathematical word problems in this class, but here you go. How many cells would be in a colony of bacteria that has a generation time of 30 minutes if the plate was incubated for 6 hours? So in my mind, I first have to ask myself, 30 minutes happens twice in 1 hour. So essentially my generation time is going to be 12. So every 30 minutes, I can count it out, 30 minutes, 1 hour, 1.5 hours, 2 hours, 2.5 hours, 3 hours, and so on. And then I would know there's 12 generations. So figured out 12 generations. So we can count that if we want to. 0 makes 2, and 4, and 8, and so on. So I can do it that way. Sometimes I do, just go back to basic finger math. But a nicer way to do this is take 2, which we know that's to double. And then we take that to the exponent of the number of generations. So that's 12. So that would tell us that you would expect 4,096 cells to be in that particular colony after 6 hours. All right, so then we can ask ourselves another question, thinking about what if we let the plate incubate for 24 hours? Now again, we're thinking about 30-minute doubling times. So you're probably already thinking about it. How many 30-minute time slots is there in 24 hours? And the answer would be 48. So now we just say 2 to the 48th power and take a look at that number. Obviously, it's easier to say it is 2.8 times 10 to the 14, but it gives you an idea. Here we are after just 6 hours, and we multiply that 6 hours by 4 more times, if you will, and now look how much we have increased the number of cells. So this is illustrating exponential growth curves. Okay, so then if we take a look at a sample of bacteria over time, we can take a look at generally the behavior, if you will, of the bacteria. So let's say you're in lab and I've given you a culture and said, hey, would you mind, transferring this bacterium from this agar slant into some nutrient broth. So that nutrient broth essentially is all the nutrients that would be in nutrient agar, but minus the agar, minus that solidifying agent. So it literally just looks like a pale yellow broth. Okay, so I ask you to do that. So you're doing all the proper things. You're flaming open the original culture. You're flaming open this broth tube. You've got your inoculating loop. It's nice and fired up, so you've desiccated anything on that. And when you're ready, then it's cooled off a little bit. You're going to, take a small amount of that bacteria colony or a sample of bacteria from the original agar slant, and then you're going to put it into the nutrient broth. So I do that by kind of wiggling the inoculating loop a little bit, and then of course I take out my inoculating loop. I want to flame closed my new broth sample and flame close my original slant, and then I'm going to incinerate any bacteria that's still on my inoculating loop. So I'm all done. Well, as you might imagine, anytime that we are starting a new population of something, even if that's just a plant maybe you got for, a special occasion, maybe Easter lilies. So sometimes we get that at Easter, we get it at Mother's Day. And when you transplant that plant into the ground, you might expect that sometimes it takes a little time to get used to its new environment. Same thing with putting plants in an aquarium. So the bacteria in that original sample, so not original, but in our new broth culture, it's going to go through a stage where it's just not really doing much. You might think of it as being a little bit in shock, or essentially just getting used to or acclimating to the environment. But once the cells are acclimated, then they're going to basically start growing. Now, that original sample wouldn't have had, what's the biggest number I can think of? Million, billion, I don't know. I always say gazillion. But anyway, that original sample that we put into our new broth wouldn't have that many cells, right? So we're thinking, well, maybe we put 1,000 in there. So once it starts growing, now it's going to be doubling every generation. And so it goes into that exponential growth phase. Well, at some point in time, it essentially is going to go into what we call stationary phase, and that's when the population has pretty much maxed out at carrying capacity. So really not a lot more room for an increased population. So there is some cell-cell dynamics. Essentially, the bacteria go into a static phase or just kind of hanging out, not dividing and just hanging out there. And then essentially that population crashes, and we call that the death phase.